Our today's topic is embryo, how become multicellular. A plant embryo becomes multicellular through a process called embryogenesis. Embryogenesis involves cell division, cell differentiation, and establishment of architectural features. After fertilization, the first cell division cleaves the fertilized egg into two daughter cells, and this is followed by a series of mitotic divisions. Cell differentiation allows cells to specialize and perform specific functions. Early in embryogenesis, essential architectural features like body axis and major tissue layers are established. Next is establishment of multicellular organization. After fertilization, the nucleus of the zygote moves to the apical pole and the zygote divides asymmetrically to produce two cells, a small apical cell and larger basal cell. Apical cell will generate the entire embryo, while the parts like suspensor, hypophysis, root cap are derived from the larger basal cell. Cell division in these two lineages produce the 2 to 4 cell stage embryo at approximately 48 hours after pollination, the 8 cell embryo at 60 hours after pollination, the 16 cell dermatogen embryo at 66 hours after pollination, the 32 cell globular embryo at 72 hours after pollination, and approximately 500 cell heart stage embryo at 120 hours after pollination. The epidermal lineage is specified at the dermatogen stage. You can see here the different stages of embryo development: zygote, then 2 cell stage apical and basal cell, 2 to 4 cells, and dermatogen. This is dermatogen stage. And epidermal lineage is specified at dermatogen stage, while ground tissues and vascular tissues lineages, as well as shoot apical meristem (SAM) and root apical meristem (RAM) are produced at the globular stage. So this is globular stage. Then cotyledons. Cotyledons are produced at the heart stage. So this is the heart stage. After pattern formation, the embryo undergoes a period of morphogenesis, where growth of the organs that were established by the heart stage lead to an embryo with fully organized shoot apical meristem, root apical meristem, hypocotyle, and elongated cotyledon. So this is bent stage, and this is mature stage of the embryo. So maturation stages include the mid-heart torpedo stage, bent cotyledon stage, mature green embryo stage, and dry seed. During early maturation, that is seed filling, embryos experience rapid cellular division and expansion, turning green and starting at the mid-heart stage at this stage, and also produce and accumulate seed storage protein at this stage, means heart stage. Starting at the early torpedo stage, the oils and waxes that form the cuticle of the epidermis are produced. Storage of lipids, carbohydrates, and fatty acids during late embryogenesis is critical for supporting the subsequent energy demand of germination. So late maturation also involves the loss of water, means desiccation, and establishment of seed dormancy. Coincident with the development of embryo, the endosperm is developed. So the endosperm is initially a cenocyte formed by nuclear division and migration of the nuclei from the micropylar region toward the chalazial end of the seed. You can see here the different stages of endosperm development in the case of Arabidopsis. So this is the condition 4 days after pollination, that is cenocytic condition. 5 days after pollination, cell aeration and differentiation. 8 days after pollination, cell expansion and differentiation. 18 days after pollination, programmed cell death. And you can see here, this is mature green stage of the embryo. So endosperm of Arabidopsis has 3 regions that become distinct as the seed grows: the micropylar endosperm here. This is micropylar endosperm that surround the embryo. Then peripheral endosperm. This is peripheral endosperm. So in the central region, and the chilazal endosperm. This is chilazal endosperm. Beginning at the third stage of embryogenesis, approximately 5 days after pollination, the endosperm cellularize. You can see here, as I already told you. So, as in many dicots, the Arabidopsis endosperm is consumed during seed maturation, leaving only a peripheral aileron-like cell layer next to the seed coat surrounding the mature embryo. Seed coat means testa, the outermost compartment of the seed that protects the embryo is the maternal tissue and consists of 5 cell layers. The innermost endothelial layer, followed by 2 cell layer of inner integument and 2 more cell layers of the outer integument. During the first week after fertilization in Arabidopsis, the endothelial cell synthesizes proanthocyanin, a flavonoid compound that subsequently oxidizes, giving a brown color to the seed coat. The inner integument layers do not appear to differentiate further, while the outer integument layers accumulate starch containing amyloplast during growth. The epidermal layer of the outer integument synthesizes and secretes mucilase. Mucilase is a pectinaceous carbohydrate and also develop a thick secondary cell wall. During the later stage of seed development, the cells of all seed coat layers die except for the epidermis. Epidermal cells develop secondary cell wall that are assumed to provide spore protection and impermeability to water and oxygen. Now I will tell you about the embryogenesis in monocot model species. Zygote of grasses like maize, rice, and wheat are also polarized with the nucleus at the apical pole. Unlike Arabidopsis, the zygote does not elongate after fertilization in the case of monocots. In rice, maize, and wheat, the first transverse division of the zygote form a two-cell embryo. You can see here the different stages of embryo development in the case of monocots. So here, this is two-cell embryo having apical cell and basal cell. Basal cell transforms into large vesicular cell, and the apical cell, this part, gives rise to the quadrant, then octant, and dermatogen stages, followed by a transition stage embryo. So this is transition stage embryo. The shoot apical meristem develops on the addaxial side while produce several embryonic leaves during seed development. While differentiation of root apical meristem defines the basal pole of the embryo. At the transition stage, this is transition stage, corresponding to about 8 days after pollination in maize and 4 days after pollination in rice. The addaxial abaxial axis of the embryo becomes obvious. As the coleoptile, this is coleoptile, primordium begins to protrude from the addaxial, means upper region of the embryo, while the scutellum, this is scutellum, arises from the abaxial side, that is lower side. In rice, the embryo reaches its mature shape when organ differentiation is complete at the 7 to 8 days after pollination. In grasses, the early vegetative stages of the embryonic seedling are incorporated into embryo before dormancy. The shoot apical meristem, you can see here, red in color, this red part, it develops on the addaxial side and will produce several embryonic leaves during seed development, and while differentiation of RM, that is root apical meristem here, lower part, it defines the basal pole of the embryo. In rice, as I already told you, embryo reaches its mature shape when organ differentiation is complete at 7 to 8 days. Although morphogenesis continues as the embryo enlarges and shoot and root meristem are completely surrounded by protective coleoptile and coleorhiza respectively. I can see here this is shoot apical meristem is surrounded surrounded by coleoptile layer. So this is coleoptile, and ram is means root apical meristem is surrounded by coleorhiza. These are the protective coverings. So at this stage, you can see clearly the shoot apical meristem, coleoptile, and this is root apical meristem, and this is coleorhiza. Thank you.